I've always been drawn to places like this. The strange part is, I wasn't raised here. I grew up in the city. Yet whenever I find myself standing on a quiet shoreline, something changes. My thoughts slow down, the noise fades, it feels familiar. For years, I assumed it was just a preference, and then I started asking a different question. How much of who we are is learned, and how much actually arrives with us? For generations, we've argued about nature versus nurture, genes versus experience, biology versus environment. But what if we've been asking the wrong question all along? Because maybe our lives aren't shaped by one or the other. Maybe they're actually shaped by the constant conversation between the two. If that's true, what happens when systems built for the average person meets minds that were never average to begin with?
Think about how quickly you lose patience in a loud room, or how easily you can maintain focus on a difficult task. Where do those baseline responses actually come from? Are you entirely the product of the lessons you learned, or are you executing a set of inherited biological instructions? For decades, science treated those two options as opposing forces. You are either shaped by your environment, or you are hardwired by your DNA. They were treated as two separate teams locked in a zero-sum boxing match. But mapping the human genome collapsed that binary. The division between nature and nurture fell apart when geneticists realized human hardware is deeply, constantly integrated with its environment. This integrated genetic architecture explains why rigid, standardized interventions work for a large segment of the population, yet generate catastrophic friction for others. If you ask behavioral geneticists to weigh in on the classic debate, they will tell you the nature versus nurture argument is obsolete. The scientific consensus has moved past trying to decide which distinct force wins. Complex behaviors are polygenic, influenced by thousands of genetic variants. DNA acts in a loop, switched on and off by environmental inputs. Genes don't write our destiny. They calibrate our baseline hardware, dictating our starting physiological thresholds, our threat sensitivity, impulsivity, and tolerance for ambiguity. But it is crucial to establish the limits of this science. Using polygenic scores from genome-wide association studies to predict an individual's specific life trajectory is an emerging field, not a deterministic tool. Consider the Twins Early Development Study, or TEDS, which has followed thousands of siblings for over 20 years. Their research found that genetic and environmental predictions currently only account for about 2% of the variance in externalizing behaviors. Outward facing challenges like conduct problems, aggression, or hyperactivity. Genetics don't guarantee specific outcomes, but they do dictate how a person's nervous system initially receives and processes the environment. We can see the consequences of that biology clearly when we look at our standard mass education models. This chart maps the reality of polygenic traits. Because behaviors are polygenic, they are not uniform across a population. They distribute on a mathematical bell curve. Here we've plotted executive function and stress reactivity, isolating the middle 68%, the statistical average. Almost all of our math systems, from public school curricula to corporate human resource structures, are engineered for that single static midpoint. That rigid design strands individuals sitting on the slopes of the curve, leaving them outside the functional parameters of the system. When an entire framework is built to serve the exact statistical midpoint, it is built for a ghost. It creates an environment that generates friction for any out of distribution nervous system. To understand the biological stakes of this friction, we can look at how we evaluate standard behavioral interventions. The textbook first then principle dictates a timeline. First, complete a low interest task like a math worksheet, and then you receive a reward like iPad time. We have to look at the exact neurological mechanics of why this holds together, or why it fails. This illustrates the physiological divide. On the left, a neurotypical baseline initiates standard reward learning. The steps remain logical and the timeline holds. On the right, a hypervigilant nervous system sees the reward cue and triggers a massive dopamine spike. Intense motivational circuitry locks onto the cue, causing attentional blindness. Because of this hyperfixation, the first task becomes a hostile threat barrier. The system demands the brain quiet itself before processing data, triggering emergency sympathetic arousal, fight or flight. Recognizing this hardware limitation has led modern practitioners to an alternative model, homeostatic plasticity. Instead of forcing an abrupt cognitive shift, homeostatic plasticity requires keeping the nervous system settled inside a regulated environment, their baseline happy place. By slowly and iteratively sliding micro steps of learning into that state of safety, you preserve the autonomic set point. You bypass the system's defensive radar. The traditional first then method doesn't fail because the individual is stubborn. It fails because the system demands cognitive hardware the brain does not currently possess, triggering a defensive override. This brings us to how humans actually operate when they aren't confined to forced institutional structures. Decades of behavioral genetics reveal a phenomenon known as active gene environment correlation, or niche picking. This outlines the core of niche picking. Individuals actively seek, create, and transform environments to match their genetic tendencies. A brain wired for high novelty seeking gravitates toward chaotic, high stimulus environments, while one with low ambiguity tolerance purposefully builds a quiet, regulated space. Human behavior is a quest for biological regulation. Our genetics drive us to select and construct the environments where our specific nervous systems can survive. We can now look back at the physical spaces we build for our population with a specific biological lens. The systemic friction in these rooms occurs because the institution assumes an average learner will sit in that chair. When the actual biology does not match that assumption, the machinery jams. Evaluating this through behavioral genetics is never an exercise in pathologizing an individual or labeling a non average brain as broken. The objective is mapping precise functional needs. It is about learning to accurately read the hardware. Human beings actively shape their environments, just as their environments shape them. That is the biological loop. Recognizing this loop gives us a clear operational mandate. We can stop forcing non average nervous systems into average blueprints and start engineering environments that bridge the cognitive chasm.